But the thing that when I was originally considering writing Poly, I was looking at what compiles to a binary, what's easy to learn, what's fast, and what has a good DevOps. It pretty much came down to like Rust was a little faster, but Go won on everything else. And that's kind of what I needed. I don't know if you've ever done like string manipulation in Rust, but it's hard. It's not something you want to teach to people coming from Python. And maybe there's a good reason for that, but I did not personally enjoy it. So when I was testing those two, it came down to Go winning on almost everything.
This episode is brought to you by SourceGraph. SourceGraph is universal code search that lets you move fast, even in big code bases. Here's CTO and co-founder Byung Lu explaining how SourceGraph helps you to get into that ideal state of flow in coding.
The ideal state of software development is really being in that state of flow. It's that state where all the relevant context and information that you need to build whatever feature or bug that you're focused on building or fixing at the moment, that's all readily available. Now the question is, how do you get into that state where you don't know anything about the code necessarily that you're going to modify? That's where SourceGraph comes in. And so what you do with SourceGraph is you jump into SourceGraph. It provides a single portal into that universal code. You search for the string literal, the pattern, whatever it is you're looking for. You dive right into the specific part of code that you want to understand. And then you have all these code navigation capabilities, jump to definition, find references that work across repository boundaries, that work without having to clone the code to your local machine and set up and mess around with editor config and all that. Everything is just designed to be seamless and to aid in that task of code spelunking or source diving. And once you've acquired that understanding, then you can hop back in your editor, dive right back into that flow state of, hey, all the information I need is readily accessible. Let me just focus on writing the code that implements the feature or fixes the bug that I'm working on.
All right, learn more at sourcegraph.com and also check out their bi-monthly virtual series called DevTool Time, covering all things DevTools at sourcegraph.com/DevToolTime.
Let's do it. It's go time.
Welcome to Go Time, your source for diverse discussions from all around the Go community. We record live each and every Tuesday at 3 p.m. U.S. Eastern. Subscribe at youtube.com/changelog to be notified, and join the Go Time FM channel of Go for Slack to chat along with us. Special thanks to our partners at Fastly for delivering Go Time super fast all around the world, and to Fly.io. Deploy your app servers close to your users. Learn more at Fly.io. Okay, here we go.
Hey Ian, how are you doing?
Doing great. How about you?
I am doing well. I'm excited about our episode today. We're going to talk about Go in biology and medicine. And our guest is Tim Stiles. Hi.
Hey, how's it going?
Great. We are very excited that you're here to talk with us about something that I personally did not do enough since high school.
Huh. This is great. I've taught a lot of people this, so this is wonderful. We'll start from I don't have any slides, I don't have any whiteboards, but I'll try my best.
So, Tim, you are doing biology with Go. How did that happen? Why? Tell us everything.
So I guess we'll start with the short story, then you can start asking me like, how did you even become a software engineer in biology? But the starting with Go was, I've been writing biology software for a while, and I'd come up on this new project where it was version control system for cell lines. So think of it as like Git for like a cell, and you're programming a cell to do a new thing, and you sequence the thing, and the cell may or not do it, and you try to track all the changes you made across these cell lines as you engineer them. And I actually went into like the original Git code to learn this, and I've written Git servers before, so for me it was kind of familiar territory. But the thing was that Python it's just-in-time compiled language. It's not really built for command line tools. And I was like, well, what is? And it kind of came down to Go, which I had played around a little bit before in undergrad a few years before, and Rust. And pretty much my decision was, well, Go is about as fast and much easier to use, so I'm going to go with that. And so that's sort of how I kind of started with that in biology. And then it kind of spun into something more, because at that time I was getting kind of tired of biotech software, because it's a hard topic. You have to know how to write software, and you need to know how to do biology, and the intersection is very, very small. And people will tell you this all across the industry. It's like the kind of thing where, like, I was in a meetup last night in biotech software, where everyone was like, why do the biologists not know how to code? Why do the programmers don't know how to do biology? And I'm like, yo, I'm right here, guys. Like, don't forget about me, because like, maybe one of a handful that can do this. And so what happened was I was kind of getting ready to leave for tech because I was kind of burnt out on this lack of tooling. And a friend of mine who was working for a professor at Stanford, he called me on his drive down from Stanford to LA where his parents lived, because he was quitting his job at Stanford. He was only like 20 at the time. He was quitting his job at Stanford, and he was just going back to like live with his parents while he started like this idea for a startup. And he's like, hey, Tim, before I leave my job at Stanford, I need to take all of these genetic parts that people have designed, and I have to take them from the JSON we have internally and turn them back into GenBank, which is this weird esoteric like sequence data format that we've been using since 1978, and it's like the sequence data format. It's like, this is what the government ships. Like the NIH and various agencies in Europe and Japan have this consortium of groups that like they have their own, I guess you could kind of call it like a GitHub for DNA, or like DNA parts or sequences. It's not quite the same. It's a little bit more esoteric and bureaucratic, but it essentially functions in a similar way. And so there's this really esoteric, designed in 1978 pre-even XML format that's all white space based. And he's like, hey, I can't convert this JSON to GenBank and GenBank to JSON and back. Like, I can't do it. I was like, you've got to be kidding me. This is the world's most common data interchange format, JSON, and the world's most common DNA sequencing format, GenBank, and you can't find a reliable way between the two? And he's like, no, I can't.
Wait, in any programming language or not in Go?
He was trying to do it in Python, and the thing that was happening is that he kept getting this like fatal error, like on several sequences where like instead of going, letting you just handle the error yourself, it would just kill the whole run. And so like the run would take forever, and he'd be like 6 hours in and be like, I don't like the specific sequence, no. Or I don't like the specific metadata, no. And it would kill the whole run. And like at that time, he's a much better programmer now. He's a great programmer. He spent a lot of time developing this with me. He's more of the biologist side, but now he's learned a lot of software engineering working with me, and it's turned into this real project where like I announced I made a GenBank parser, and people were into it. It was like a thing. Like I was like, hey, I'm done with this. It took me 3 weeks of going through various poor forms of documentation. I found one European website I think is official from the government that had bad SSL certificates. So like it gave me warnings when I tried to click on it in Firefox, like don't go to this bug infested site from the government. And I was like, okay. And like I got to it, and that's the only specification I could find for the file format. It took me 3 weeks to write this parser. It's not like I haven't written a parser before. I've written plenty of web scrapers in my lifetime. It's not like it's new to me, but it became this real thing where I tell people often for biotech, it's a big deal if you put it in JSON, because they've been handling data formats since before the public, think Usenet era, 1978. Like, we don't even know who made the most, maybe we can find out who came up with JSON, but GenBank, like, I've tried to figure out who came up with GenBank. I can't. All I know is there's some council of elders in 1978 got together in the deserts of New Mexico and decided on this format. And there's no record of who these people were. And we've all been living with the consequences ever since.
This is really mind-blowing. I had no idea, and wow.
It starts with this complexity of this, what you call just legacy data or legacy code or software, because, bioinformatics has existed since before the web. We've definitely evolved a lot since the internet came about, but scientists have been using the internet before everyone else by at least 15 years maybe? So there's a lot to parse there. So the most recent file format that I've seen GenBank, the NIH-backed, United States NIH-backed database use, is something called ASN1, which is like the precursor to XML. It's like what XML was to JSON, but for XML. It's super weird. And someone asked me, have you ever written a parser for this? I'm like, I didn't even know this was a thing. This is amazing. But also, no, I never want to do this. And so a lot of biotech software is limited by the fact that there's these data formats that the government uses, or, some repository uses, but everyone else is just sort of most people doing this work are scientists. They're not software engineers. And there's a big push right now for scientists to learn a little bit of software engineering, DevOps practices. I'll make a post this is what unit testing is. Have you ever heard of example tests? They're really great in Go. And they'll be like, you saved my life. I can actually write my code. It's really kind of, it's kind of wild. So there's this whole discussion about right now in the field of how do we write better software? And I've kind of find myself in the center of it because I wanted to write this to make cool stuff. It does have medical applications, but I'm thinking of stuff like flying seaweed and Tide Pod trees and all this other goofy stuff that I think of in my spare time where I'm like, someday I'm going to need the software to do that. But it just didn't exist. And the tools that you mostly find are usually either, old C or Python packages, or there are companies that rewrap it in a nice GUI and sell it to scientists for a good price. And, they do that, but it's not really a programmatic approach. You can't do much with it other than drag and drop. So for example, DNA synthesis, which is what we use to make new DNA for like certain genes that we're trying to engineer, there's only like 2 companies that have an API for that out of like the dozens that exist. Most of the time, if you want to like send something to them, you have to do like a drag and drop into their GUI. And I talked to a lot of vendors about this, and it's just like, yeah, there's only 2 or 3 I know of that have an API. One of them you have to send your DNA in Excel format. They don't take JSON, it's Excel format. I know, wild. But yeah, I can get off this diatribe for now, but yeah, it feels a little wild.
So that's kind of the tech side of things. How'd you get into the biology side of things? I think before the show, you said you had a degree in computer science, but how'd the biology come in?
That's true. So this is really funny. So back when I was a student, I first started as a mechanical engineering student, and then I switched to it as a design student, and then I got really into robotics. And I was playing around with 3D printers, and I was doing a lot of computer vision stuff, and that was kind of like my jam for most of undergrad, was doing computer vision stuff. And at the time, the NSF here in the United States was giving out grants to undergraduates to, do computational bioinformatics biology research. And so I was like, well, I like money and I want to get my undergrad paid for. So I found a couple of professors to work with. I tried out a few things, and I kind of stuck to computer vision until I got this job that kind of bridged out from undergrad, where I was working at Harvard Med as a research assistant doing computer vision for endothelial cell morphology, which is a fancy term for the cells that line your blood vessels, how they move and shape and morph as they respond to hypoxic cells. So if you have a cell that is like, doesn't have enough oxygen, it releases, something called vascular endothelial growth factor, VEGF, which is a chemical that goes through, all the nice little fluids and tissues in your arm or your body. And when it hits the endothelial cell goes, hey, that cell's in trouble. And it coordinates all the other nearby endothelial cells to shift and morph and change their, the rigidity of their cell membranes to sort of push their way towards this hypoxic cell to deliver oxygen. And the reason why this is important for study, obviously it's just cool first off, but the reason why a lot of people like to fund it is this is a dynamic that's very central to tumor growth and cancer research. Because what tumors do and what cancerous cells do is they put out this VEGF factor to an extreme where they're just saying they're constantly hypoxic, even though they're not. They're saying they're constantly hypoxic and recruiting all these endothelial cells to feed it more and more blood, essentially, to deliver oxygen to it and let it grow rapidly. So usually the line of research there is, can we find a way to mess with that to stymie tumor growth? And the answer is, yeah, a little bit. But, cancer is a very complex disease with lots of different ways of presenting itself, and it's not the end all there. So I got started in that, but at the same time, my spouse, Ren, we were dating then, but now we were married. She was working at a lab at Harvard Med under George Church, who is a famous synthetic biologist. He's like in Wired magazine. He's a really nice guy. He says yes to everything. So like if you have a synthetic biology startup, he'll be on your board. But, I don't know if he'll have enough time for you, but he will. And so she was working in his lab doing cell fate programming, which is this concept. Have you ever heard of stem cells? Do you know what stem cells are? Mm-hmm. So stem cells, the idea is that they're kind of, cell fate programming is the idea that you can turn a stem cell into another kind of cell, a blood cell, a tissue cell, an eye cell, a different cell. And you do this by introducing various chemicals to it and like these different stages. So you're programming with some various, enzyme or hormone or some odd factor, and it becomes a different kind of cell. And a big thing that people have been trying to do is figure out how to take cells that have gone from stem cell to fibroblast cell or skin cell and then turn it back into a stem cell. Because if you can figure out how to do that and culture those, it has a lot of applications for, say, tissue engineering and replacing, degenerated tissues and things like that. It's still all highly, I want to say speculative, but it's new. It's like we've been doing it for a while. Maybe there's a few therapies for it, but I'm actually not a medical guy. I'm a little squeamish, which is hilarious to a lot of people, but it's true. If I'm not familiar with it, I may get a little nauseous talking about it. But to say how I got into biology is that eventually doing this, walking home with my wife, we were like in the same area. So, I'd walk 15 minutes to her office and it was on the way home and we'd walk home together and she'd be explaining her work to me. And she was mentioning this thing called plasmids, which I can talk about a little bit more later. But essentially, they're these little circular tokens of DNA, these little, consider them like tiny functions that contain like a gene, and maybe some things to promote the expression of this gene, and a couple of other things like maybe some sort of resistance to an antibiotic, which is useful in lab plating. It's called plate selection. I can talk about that too, but essentially it contains like 2 or 3 genes. It's like 10,000 base pairs, which is tiny compared to all of your genome. But they occur in nature and bacteria as a way of getting around the fact that they reproduce asexually and they need some genetic variation. But we figured out how to use them in other places to do sort of like small genetic testing. So she would call, they're like tokens, but she would call them circular. And I was like, wait, when you project it, isn't the helix kind of circular when you look at it from a certain projection? She's like, no. And so I was having a hard time. There was some problem with staining where like I couldn't get quite the information I wanted out of the data I had. There was something that going on there. It's been so long since I looked at this, but essentially I was like, I need to be able to explain what colors I need and how it works for staining. And, I can't rely just on the wet lab biologist to kind of bridge this gap for me. It's not going to work that way. And so eventually she got tired of explaining things to me and very gently suggested I go to this garage lab in Somerville, Massachusetts called Boss Lab, which has been around forever in some various way, shape, or form. And I showed up there and I was like, hey, I need to learn biology for my work. And they're like, yeah, we were going to make a class, but we haven't done that yet. So if you teach the class, we'll teach you how to teach the class. And so I spent I'd say 6 weeks in this lab, nights and weekends, working on this, what's called the heat shock transformation protocol to put these plasmid tokens into bacteria and kind of learn the basics of biology that way. And it was kind of like a world-class education in a sparse environment because the people who I was messaging for help were PhDs from Institut Pasteur and UC Berkeley and UW-Madison. And there's a software engineer who's the VP of engineering a data robot. And they'd all be giving me advice over Slack, oh, this is over here, this is over here. But I was kind of also on my own because none of them had made this protocol before. And from there, I kind of, just got too into it, kept getting contracts and kept doing things. And eventually, I was kind of struggling and I wanted to get out of the field. And I just kind of stumbled on something that worked. And now I make my living as a consultant doing something that works, which is really great.
You have also built Poly, which is a Go package for DNA.
Yes. So for engineering DNA, which is is very different from what a lot of other people do with DNA. A lot of times they're looking for diagnostics. They're looking for variants. They're trying to assemble it into a whole genome for reference. And what my package does is it tries to engineer DNA to be used for certain experiments and certain designs. And so what my package is mostly focused on is going from designing DNA into, as a concept into something that you can put into cells, which is surprisingly unique. You think there'd be more of this, and there are. There's a lot of companies that write this internally for themselves. It's imagine they're writing their own React framework and then not sharing it. And that's kind of what's going on in almost every company that does this. They burn millions of dollars doing this, and they don't share back in most cases. And so I wrote this as a thing where I was thinking, I want to be doing this for the next 10 years. I want a really stable library. I want it to be really well tested. And I want to have the features that I need to create cool stuff. And so I started from sort of that principle. And it started with just parsers, which every bioinformatician will tell you that's the first thing they have to do is they have to write parsers. It's the bane of our existence is taking government data and putting it into JSON. I wish I was kidding. But after you get past that, then you get into stuff well, how do you manage having a hash ID for a circular sequence? Because by circular, the string is circular. There's no way of telling where it's going to start officially. So it turns out there's this old algorithm called boost least rotation that would, given a certain sequence that's circular, will always bring it to the deterministic point. And we use that to create hash IDs for these plasmids, because otherwise it would take an inordinate amount of time to figure this out. Because then you're using pairwise alignment algorithms, and these are not linear algorithms. They're kind of gnarly and not my specialty. People actually specialize in string alignment algorithms for the specific stuff. Heng Li, who's a professor at Harvard Med, specializes in this, and his software runs the world quietly. He does not get enough credit for how much bioinformatics relies on his stuff. The practitioners know, but the people downstream have no idea that this one Harvard professor is keeping all of our, if you've ever used 23andMe, Ancestry, or anything else, or any genomic testing ever, your DNA has probably gone through one of his algorithms. He's very prolific. And unlike myself, I still have a small user community. It's I have some very hardcore users that have their own startups that are raised money for it. And, I have a handful of people that give me feedback. And I've even had large companies go, hey, we're thinking about using this. But I still feel like a small player in comparison to the people who've come before me. Again, I wrote it because I want to engineer DNA and I think it's super cool. And it was kind of hard to do that with the tools available.
So I want to kind of get into like the nitty gritty details of it. But first, can we talk about like kind of high level what it does? Like what kind of projects could you do with it? Are there projects you've used for it?
Okay.
Just kind of get some context here.
The thing is, it's a very vast library despite how much I've underplayed it so far. So some real things are, I know that a couple of consultants have actually engineered microbes as a potential therapeutic. And I've done that as a consulting gig for a couple of YC companies that I probably shouldn't name, so I will not. There's another company that's trying to automate DNA synthesis and cloning, which is like actually a big-ish business. There's plenty of companies trying to do it, and he's like a main contributor, and like they just closed a $3.5 million, or about to. I don't know actually. Blah blah blah. But if you're interested in like writing Go and doing synthetic biology, check out Trilobio in San Francisco. They're really great. I keep my surfboard there. I can recommend them wholeheartedly. But there are things that people could be doing with it that they aren't yet. One is designing primer tests, like the PCR tests, like the ones you've seen for COVID. There's very explicit tooling to be able to do that for designing primers and looking at. I'm currently working on a project where I'm essentially taking all this GenBank data that I told you about, the antiquated, GitHub of DNA, and I'm putting it into like, a graph database of all this little fun extras. And part of that is now that I could probably, hopefully, be able to design primer tests for almost every viral string of DNA we have, which is really cool. So there's a lot of medical applications. There's a lot of biomedical applications, which most people, when they talk to me, are very interested in because it's innately human. Everyone's had a medical condition or knows someone that has a medical condition. And one of the examples that I should have started with, I don't know why I didn't, is if you're diabetic or you know anyone diabetic, they are taking insulin that was produced in yeast. Not, the classic way, which is like it was originally derived from pigs. But in the 1970s, I believe it was Eli Lilly engineered yeast to produce, human compatible insulin. And then this is how synthetic biology kind of works, at least at the early stages for a lot of companies, is they engineer yeast or E. coli, not the dangerous kind, not the Chipotle grade E kind, but like lab safe, like difference between like a rabid bull and like, a cow, like a domestic cow sort of difference. And they engineer it to produce some molecule, some chemical, some drug, some valuable-esque thing. And then they bring it to, a contract brewer in Wisconsin, and they go, hey, can you take the strain and brew a ton of it, and so we can extract the thing of value? And so that's how insulin's been produced since the 70s, is this way. And it's actually part of the reason why I think it's so criminal how expensive it is, because it's actually really cheap to produce. But the thing is that this is a technology that's been around for longer than I've been alive. It's been around for 40 or 50 years, but it's sort of become this new thing in the public eye of, oh, I can do it too. It's not just the realm of Monsanto and Eli Lilly and pharmaceutical companies. It's now getting to the realm of people like me who have created this library that, other, companies use. And like, I wrote this in the comfort of my living room. Like, I haven't had access to a lab in too long. I want to build my own lab again. But like, it's at the point where now like anyone with a computer can contribute in some way, even if they don't quite understand biology.
Sorry, I'm processing for a second there.
Yeah, sorry.
Yeah, a lot of information. Super interesting.
This episode is brought to you by our friends at Square. Develop on the platform that sellers trust. Here's what you could do with Square. You could bridge more experiences. You could build online, mobile, and in-person commerce experiences that connect more customers and sellers. You can build custom booking solutions. You can create and track orders. You can accept payments. You can manage and curate inventory. You can organize customers. You can manage employees. You can extend Square gift cards to your app. You can use Afterpay. And all this is powered by the world-class Square APIs and SDKs that enable you to build full-featured business apps for yourself or millions of Square sellers. So much is available as a Square Solutions partner. Learn more and get started at changelog.com/square. Again, changelog.com/square.
So that kind of made sense to my limited mind, right? But so like, what does it do? Like, are you like running simulations? Is it statistical modeling? Is it like how DNA reproduce? Like,
Not statistical modeling, it's more construction.
okay.
So think of it this way. If you need to engineer a DNA construct, like these little plasmid tokens I've told you about, these little, essentially swappable functions we like to put into different cell types, you have to construct that. They exist in nature as bacteria, but for our purposes, we need to make sure that it's human compatible, that it'll express the gene and all this other stuff. And some people, they take essentially, templates where they just swap out the gene for a different thing and it'll, express in the organism of choice. But my software makes it easy to design thousands of these at a time. And traditionally, the way that most people do this is they design it one at a time with like a CAD-like GUI software. So say you want to run a whole DNA synthesis operation, my software does that end to end. You can design the software, you can design the construct of interest. You can simulate how you're going to construct it. You're going to simulate there's special ways of printing DNA. So DNA synthesis is a little funny in that there's some sequences we can't print, but we can print analog sequences that are similar enough it doesn't matter. But we have to know that. And so my software, for example, fixes that little quirk. It optimizes to be expressed in the cell because different strings of DNA will be expressed differently in different organisms. Like they'll express in both, but at different rates, depending on like usages of what we call codons, which I should have just started with a lecture on the central dogma, but you can look it up at home. It's called the central dogma of biology if you're listening now. It's like the core tenet of like how like the biology you need to understand this. So essentially, my focus was just on making it so you could engineer a DNA string to do what you want and it would express faithfully, and you wouldn't have problems when you send it to a DNA synthesis vendor with them going, oh, it's not the right sequence. And so you could do it at scale instead of like, one at a time via one of these mini CAD-like tools. You could engineer thousands at a time, and which is what a lot of scientists do. I'm not sure how they do it. I think they're mostly also writing Python, which is not fun for them because they're writing a lot of this from scratch. But that's kind of like the method of production here is you either use CAD software and you write one or two, or maybe you can get more out of it, or you write a script that generates thousands of primers for, say, PCR tests that you're trying to develop, or thousands of variants of a plasmid, these little tokens I've been talking about that will, express something for an experiment, or you have to do it one at a time. And so my software is really the only software out there that's fast and stable and can do this reliably with like high unit test coverage. There are, I would call them predecessors that are really impressive. And there's also really impressive GUI tools that you'll do this one at a time sort of thing too that are open source. My project is like the first that makes it really possible to do this at a scale where you're not looking at bad compute bills. This is the thing is like synthetic biology shouldn't have high compute bills. It's not like we're looking at whole genomes all the time, unless you're working on human specific stuff, which not everyone in synthetic biology is. A lot of people are, obviously, because that's where a lot of the money is. But the strings you're working with are on the order of 10,000 kilobase pairs is what we call it instead of kilobytes, which is 10,000 letters to 4 million, I think is what E. coli can be, and 10 million maybe for yeast. I forget. Some biologists out there, if they're listening, is probably screaming at me for getting this number wrong. But it also varies between yeast strains. And so I'm running out of words, so I think I'll let you guide the topic to something.
Yeah. Could I repeat that back to you and just see if
Yeah, of course. This is a biology lesson for everybody. I wish I was talking more about Go. My practice is there.
Oh, well.
Should have done that too, but yeah.
We'll do Go right after this, I promise.
Okay.
So basically, someone has engineered this plasmid that they want to be able to produce, right?
Yep.
To do whatever X thing, make a new protein, whatever.
They've literally just whiteboarded it. It's a concept on a napkin.
Okay, yeah. And so they take your software, or this is one thing it can do at least, is run simulations to try to find actual DNA that we can produce.
Yes.
Synthesize in real world.
Yes.
But we don't have to touch the real world. We can just simulate it and yours can be
exactly.
Yeah, we can make this. Okay. That's cool. Okay.
It's really valuable because each one of those plasmids I've been talking about, the price is always going down for DNA synthesis. But right now, it's not uncommon to hear of a $500 to $700 bill per plasmid. It's not uncommon. I have a customer who, I'm working with right now, and they make DNA libraries that every time they send out an order, it's $20,000 to $40,000. So for them, it's worth it if they have a little extra confidence that instead of, getting their grad student or their, a lot of scientists don't know how to hire programmers. I can get into that dynamic too a little bit. It's a really fascinating one. But essentially, instead of having to worry about, will his DNA synthesize and all this other stuff and maybe blowing $30,000 out of his research budget, he just comes to me and says, hey, can you write a pipeline for this out of the software that you wrote? And I go, yeah, I can do that. And, it's cheaper than what it would cost him with his own, hiring his own people. And it's just a nice deal all around. And he gets to feel oh, this is secure. There's testing behind this. We'll have like, because a lot of scientists, it's a lot of prayer. I mean, like if you ask a scientist like what they do before they send out a sample to be, sequenced or go through cryo-EM, or before they, they try to collect data. There's this false dichotomy between religion and science, but a lot of them do a little, even if they aren't religious, do a little prayer like, I hope this works, because scientists are engineers. It's a difference in practice. Scientists are trying to discover something new, and they're doing it like the startup-y, like, got to do it as fast as possible, plant the flag, get the research, get the data, boom. And engineers, we can be a little slower, we can be a little bit more pragmatic, we can make a little bit more stable sort of maintenance conscious decisions. And I think that's sort of what my role is with scientists, is I make these maintenance conscious decisions where they can go, oh, that's a good library. I can really trust it. I can really believe in it. I know that Tim isn't trying to run as fast as possible towards this goal. He's trying to build for the long term.
That is really interesting. We talked about what it is, what led to that, what it is doing now. Tell us about the future of bioengineering as you see it.
So this is a conversation I come into a lot. And actually, I have like friends who we are like on polar opposites. It's like one of those things where we both agree where the future is going, but we disagree on how the future is going to get there and like what the future, like the practice of it's going to look like. So for me, the practice of it in my mind is, software engineers who have a decent enough biological experience. It's easy to learn, hard to master, all the theory behind it. And, it's a lot of little tiny variables, which is computer scientists and engineers. We know, we have lots of tiny little variables all over our code. We're aware of this. We have to keep tracks of lots of little tiny details. But there's this whole breadth of research, and every once in a while, I get dragged into a field that's adjacent that I know nothing. Like I had to learn about immunology recently. Immunology is like its own thing, where it's heavily related to genetic engineering practice, especially with these new immunotherapies we're doing for cancer and stuff. But I had to go and learn about immunology as a biological basis in human medicine. But the thing is that we're going to get to a point where hopefully there's enough software engineers with biology experience that can talk to these people that have this experience in more pointed niche parts of the field, like immunology or plant physiology or these different, biological topics where they can work together. And someone will say, hey, I need to go probe this. And, they'll talk to the engineer, and the engineer will get enough that they can work with them. And that's what I would like to see is, two scientists and 20 software engineers writing some awesome code doing stuff. And then my friend is entirely opposite. He thinks we should have a few software engineers writing not GUIs, but kind of like that, report generators maybe, and it's just 20 scientists analyzing them, and we have entirely different outlooks on how it's going to be. So what I'm betting on is that we're going to have in the future, we're going to have a lot of software engineers coming from languages like Go and Rust coming into biology and sort of speeding things up. One of the things that's fun is that every time I rewrite an algorithm from Python into Go, it's 25 times faster by default. We have the world's fastest DNA synthesis fixing function by several orders of magnitude, which is kind of nuts. We weren't aiming for that. That's just kind of how it happened. So there's a lot of work to be done there. And I think the real impetus for most software engineers is the biology is intimidating. And I think the one thing, if there's a software engineer listening to this right now who's interested in biology, don't be so intimidated by the biology. Seek to learn it and be humble when you ask questions to scientists, because they've gotten a lot of physicists and software engineers over the years coming into the field and acting cocky because they see, this code that they've been working with and they're like, oh, these guys don't know how to code. They don't know nothing. It's oh, no, they know something. It's just they're not software engineers. And you really got to listen to them. You got to be humble, and you can't come in with this attitude that you're going to solve all their problems, because you're not. My favorite is, do you guys know the three-body problem? This classic problem in physics has been around forever.
Yeah.
Where essentially we don't have the math to model three bodies orbiting each other, all related to each other. We just don't have the math for it. And so that's what the protein folding problem is. That's why everyone gets excited when there's a new, people here probably have heard of AlphaFold here. Maybe you haven't, but every time AlphaFold comes up with something new, it gets really exciting because they're fighting the three-body problem with huge gobs of data and models. But I've had mathematicians and software engineers go, oh no, physicists would be like, oh, that's a simple heat model, and spend 45 minutes trying to explain to 5 biologists why they're wrong and it's easy. And I'm like, no, we all know dude, we took freshman physics too. We know what the three-body problem is. Stop. That's the one piece of advice if there's a software engineer who wants to get in genetic engineering or DNA synthesis or anything out there, is just remember to be humble, because you'll learn something new literally every day and you'll be like, wow, this should be some basic stuff. But that's kind of how it goes when it this broad layer of basic stuff, and then you get into really fine details.
So you mentioned kind of Go and Rust and the future there.
Yep.
Do you think Go has like a prominent feature there, or
Yes, I do.
Why?
Usability is the biggest thing there. And also just the DevOps tools are amazing. There's nothing that makes my life easier than writing in Go. It's like, well, first off, almost every tool, every DevOps tool is written in Go now, like almost every single one. For my project, I regularly use Gitpod's a real favorite of mine personally, but
it's a German thing.
yeah, no, they're great.
Yeah.
I really love them. By the way, anyone listening out there, you haven't been to Gitpod's Discord, go. They're super friendly. Just show them your work with whatever they're doing. They're super excited and supportive and ask a lot of questions. They're really, really nice. But when I got started, Go's like test first. Like it's all the things you'd need, example tests are actually a great thing where like Python or other languages, like maybe there's a library for it, but like in Go, it's just standard. Whenever I write an example test, it runs every time I'd run my test, and it's an example that never does doc rot. The problem with doc rot with a lot of scientific software is big because people will write these huge things of documentation, then they'll change the API like 2 versions down, and then they won't change the documentation. And so I think the fact that Go has all of these opinions, which if you're like I guess you would call it like a graybeard or whitebeard software engineer, like maybe you have different opinions, but if you're just starting out and you don't really know where you're going DevOps wise, Go's defaults are beautiful. They work. And you may struggle with like generics, which Go's obviously been working on, which I think scientists will have a little bit of a hard time going to type languages. They don't have that much of a hard time. But the thing that when I was originally considering writing Poly, I was looking at what could be compiled to a binary because I thought it was going to be more of a command line tool. Eventually I was like, wait, this doesn't make any sense as a command line tool. But originally I thought it was going to be more of a command line tool. So I was like, what compiles to a binary? What's easy to learn? What's fast? And what has a good DevOps ecosystem? And it pretty much came down to Rust was a little faster, but Go won on everything else. And that's kind of what I needed. I don't know if you've ever done string manipulation in Rust, but it's hard. It's not something you want to teach to people coming from Python. And maybe there's a good reason for that, but I did not personally enjoy it. So when I was testing those two, it came down to Go winning on almost everything. And I think it's also just a great step from what would be considered these scripting languages where there's, it's still, the syntax is easy enough. The concepts are still there. I mean, yeah, you can get into concurrency and all this other stuff, but coming from R and Python, you have to learn that anyways. You're not learning multi-threaded stuff. And what most scientists use for languages, which are usually most scientists are using Python, are MATLAB and Julia, which actually Julia does have multi-threading, a lot of, modern features. But Go just has a bigger breadth and a bigger DevOps community and a bigger community of people that use it, which actually makes it really easy to find odd functions that I need. We use a Blake. One of my contributors convinced me that we need to use Blake 3 for circular sequence hashing as the default. And so Go's hash crypto library doesn't have that yet. So some guy wrote a working implementation and we just use that. And a lot of Lua didn't have that. So my friend who wanted to implement it in Lua had to go and write Blake 3 by hand. He's like, I don't understand this. It's all a bunch of math and single letter variables. I was like, so now do you realize why I make you write all your variables as whole words? He's like,
grumble.
That's something that some of my contributors are like, why do you make us do whole words? I'm like, you'll thank me in 6 months. And, Go has all these great tools for detecting race condition, data race conditions. And that's something I learned recently. I'm still learning various things. There's a tool where you can find unnecessary conversions. And there's code coverage tools that are just native. And it's all easy to throw into a GitHub action or any other CI/CD thing you're doing. And so for me, it's this thing where there's all these wheels already built in. And then there's community people that really care about how usable and shippable their code is, which makes it just so much easier to integrate their stuff into mine. So a lot of scientists, they heavily rely on Docker and Jupyter notebooks, super, super, super heavily. And usually they're like, what do you do to containerize your software? I was like, well, Docker's also written in Go, so I just do what they do, which is ship binaries if it's an application. And I just have a Go releaser that, script or a CI/CD thing that puts out every architecture and operating system that it possibly can. And I think the only if you find a system that can't run that, you're advanced enough that you can figure out how to package yourself. That's that's it. If I'm doing what is it, Fedora, Ubuntu, Arch Linux, you have enough. And so I just think as a programming language, it's just really powerful and really suitable for people that are trying to build something stable that may not have had that experience people in biotech have this insecurity of feeling like they're not real software engineers. That's not necessarily true. It's just sort of they're working from this corpus of work that is, as I said earlier in the show, since the 70s. There's a lot of legacy there that we're still dealing with. And part of this project is kind of jumping over the legacy and going to something faster and easier. The stuff I've written has mostly been for DNA engineering, but next I want to get into metabolic pathway engineering and protein engineering. And these, that's sort of the whole reason I've kind of gotten into this is, I want to make proteins. For those listening at home, you probably heard it in the bodybuilder terms, if you need enough protein to build muscle mass. But proteins, believe it or not, is like most of your dry weight. They're actually nanobots. Believe it or not, you are the gray goo. There's all these proteins that are coded in your DNA that are expressed all the time that do little functions for moving DNA from one part of the cell to the other to helping the cell split that are super specialized. And they even have you ever seen like a bunch of magnets when you throw them on a table and they go like that? They sort of just kind of globble all together. That's kind of how protein folding works in a little bit of a way. They're all like little molecular magnets kind of folding together and doing this thing. So what I'd like to do is I'd like to make the nanobots. I would like to, there's a lot of really interesting applications for energy, for, plant life cycles, for almost anything. I mean, they're nanobots. What can you do with these? And so I'm really excited about that, but I'm also excited about the macro stuff, the stuff that we haven't touched on. Traditionally with genetic engineering, the money makers are first pharma, then ag tech, and then other. And I'm really, really, really interested in the other part. I think that's the coolest part. Like, can we make plants as infrastructure? You may have seen the Glowing Plant Project, which is totally
like mushrooms.
Yeah, like the mushroom bricks that you may have seen. Like, why can't we just have the mushrooms grow to house size? Is there some sort of fundamental physical limit there, or have we just not tried hard enough yet? I saw a great quip on Twitter where someone was talking to a group of kids and is like, so are you working on, I think the kid asked so are you working on, can we make elephants fly? And they're like, I don't really know. And he's like, what? So you're just going to give up? And it's like, yeah, why don't we try? And I'm a little more squeamish about working with whole organisms, the kind of organisms that vegans would not want to work with. Oh my God, mice work in biology is scarring. If anyone's out there is thinking about, working with biology lab, just don't do I hate to say this because mouse work's important and it's a big part of science. And Europe actually tried to ban mouse work at one point. There was talk about it, and a bunch of Nobel Prize winners were like, I'm really sorry guys, but we can't, that would be a huge cost to society. We can't do it yet. But with plant work, for the most part, there's a lot more to be done there with working with morphology and expressing different genes. You can actually, grow plants that produce medicine, which people have been working on. My personal favorite is people really want to build infrastructure. They want to have, you may have seen on Instagram, these trees where people have guided roots across rivers for generations. It becomes a tree bridge. And it's wouldn't it be cool if we could just plant a tree and it just was oh yeah, I'm going to go over this little thing and make a bridge. That would be great. That would be super cool. And that's kind of this sort of future utopia that a lot of people in my field are looking towards, is the other stuff. But we also know that pharmaceuticals is super important. It's also a very meaningful thing because we've all had families in hospitals before. So it's something that we all respect. And, if you're squeamish like me, I try to avoid it, but also it's important. And the ag tech stuff, Monsanto ruined the whole. So the reason why we call ourselves synthetic biologists instead of genetic engineers, Monsanto, their lawyers and PR team just really messed up. I don't blame the scientists at all for what's going on, but their PR and legal team, they really messed up. And that's why we call ourselves synthetic biologists now, for everyone at home. Because we didn't want to have the same flack because we weren't doing the same stuff. So, yeah, I think that's the future of biotech, maybe. There's a lot more to it. But you'll see a lot more, specialized cancer therapies, specialized gene therapies are another thing that is probably going to be in the near future. We're already starting to have it. There's a lot of essentially what we call CAR T cell programming or reprogramming, where we take a sample of a patient. There's probably someone in immunology and CAR T cell therapy listening to this that's you're so wrong, Tim, don't get this wrong. Essentially what we do is we take the patient's cells, we take them out, their CAR T cells, we re-engineer them to target the cancerous cells that we've also sequenced, we do this in the lab. Then we reintroduce these CAR T cells to attack the cancerous cells and not the healthy cells. Because one of the issues with cancer is essentially your immune system doesn't know how to specify things in the body. I mean, there are some cancers where it figures it out, but there's a lot of, the problem with cancer is that it's your own cells. They're very close to what your regular healthy cells would be doing. So for your immune system to be hey, that's bad, it's kind of hard for it to pick up. And there's a couple of reasons why, how the adaptive immune system works, so why that has to be. But if we can take these people's immune cell, these CAR T cells, these immune cells out and engineer them to attack cancer cells without attacking that's the crux. That's the dangerous part, is if you don't engineer it right, it attacks, the healthy cells, which is not good. But the idea being that In the future, maybe we don't, there's already therapies that don't involve chemotherapy. I'm pretty sure. Again, I'm not a medical expert. This is not medical advice. Please talk to your oncologist about any cancer-related needs. Actually, that's one of the big things about being a biologist is you find yourself in this position where you're explaining biology and they're like, hey, should I do that like health-wise? I'm like, no. Go talk to a doctor. They have liability insurance. They've been trained in patient-related matters. They know what drugs do. I'm just the guy that does the biology that gives them the tools to do that. Like, that's the difference here. Like, I've gone to the doctor. I'm like, my stomach hurts. Is there something wrong with my microbiome? And they're like, no, Goofy, you have acid reflux disease. Take this Alka-Seltzer and stop eating spicy food after 8. I was like, okay, cool. And they were right. And that's kind of the thing. Like, again, when you're like being a biologist, you have to be humble. There's always going to be another expert in some specific niche that you're working in. Will
AI be one of those experts?
You can absolutely be one of those experts. Actually, that's been a joke that I've had
can artificial intelligence. Yeah.
Oh, AI. I thought you said I personally. I said you can be.
I didn't even dream of that going that direction.
No, you can do it. You can still do. I'm not kidding. I had one coworker that's like, how long would it take me to be a master in like alignment software? I was like, master, a long time. Second best, maybe third best. It depends on how fast you read, but it could be this year. It really depends on like what your focus is and what you're doing. Mm-hmm. We all have our unique specialties. But for AI is this weird thing where, machine learning is so obvious. Everyone in tech knows it's obviously overhyped, right? It's like this kind of thing where now we're having all these generative models, which are awesome. I was writing basic genetic programming, like generative models for like weird little Pokemon like a decade ago. But now we have full artistic renderings of like, for example, my Twitter banner is like a flying seaweed monster. I had to like tweak it a bunch, be like flying spaghetti monster, but with seaweed and some other stuff. And like eventually I got it to do the thing. But we're at that point where we have these generative models that it's been a long time since I've been in deep learning. So not only has the field changed a lot, but the terminology they use has changed a lot. That like every 5 years they have to reinvent every term for linear algebra. I don't know why they do this, but they do. It's a weird thing. I had a similar problem when I was first learning it like 5 years ago. I was like, I think it was, what's a linear filter? Isn't this just a kernel? And then the PhD I was working with was like, yeah, we just do that. I'm sorry. And like, that's a similar thing. Now there's all these different terms. So I'm like, oh, is this basically a convolutional neural net? And they're like, yeah, but it's different. And I'm like, okay. So with machine learning and AI, I think there's a lot of opportunity to get around what have been these traditional problems in modeling where we don't have the math for it. We don't have, like I said, with the 3-body problem, we don't have the math. No one's invented a deterministic, heavily reliant data way to, model 3 bodies orbiting each other. It's just we haven't done it yet. But David Baker, who is a professor at the University of Washington, his lab came up with this game called Fold It, where essentially they would get all these molecular biology students in college and maybe high school even to play this game where humans are actually pretty good at figuring out from like a basic string and some mechanical principles, like, a 3D model of this like DNA laid flat, how to fold it like origami into a shape that's is what we'd expect at the end. So they'd have the flat sequence and then the expected sequence they get experimentally. And they'd have these students essentially be mechanical Turks for fun to train this model to figure out how proteins fold, which is like, super useful. So in this case, there's some real roadblocks where machine learning is absolutely vital. And you do need machine learning for it. But there's also a lot of people that do machine learning models. And then you're like, you didn't need machine learning for that. It really depends. But the folding stuff and the molecular dynamics stuff, stuff that, the molecular interactions, I see a lot of potential there. There's a lot of work that can be done there, especially so there's something called ligand binding, which is like a fancy term for it. We take a small molecule, we'll make it attached to some protein of interest or something that disrupts some function that helps cure some disease. It's all downstream. Like if you tried to do like a stack trace of like how medicine works instead of bug failure, it's pretty brutal to be able to do that. We're still at the point in life where we don't really have that. The closest we get is my software, which is, that's not good enough. I'm going to be honest. Go stack trace cannot heal you. Sorry, that's not a good endorsement of the language, but we'll get there someday. But that sort of space of figuring out what molecules interact with each other and how they fit and how they puzzle piece together, that's some really good use cases for machine learning where you're just convolving 3D structures against each other. You're doing, I call it subsampling, or people call it fingerprinting, where you decide, this is the only place where we really care about seeing the molecule binds because that's the therapeutic effect. And so there's a lot of research there in that, and that's super valuable. And I don't think there's going to be many other routes to doing it. Of course, there's these classical deterministic ways, which are good shorthands, and they're probably faster. And you don't require a big beefy supercomputer to pull it off. But I think that's the place where machine learning is really that's the real strong suit there. There are things with people have been figuring out how to do DNA synthesis fixing like I do, but like with machine learning, and it's like, I don't know, mine works like 99% of the time or something. And it's like, there's no machine learning involved. And I haven't really looked at the other people's models, mostly because I don't even think they're on Hugging Face. I mean, I got to give the Hugging Face guys super props. There's also a machine learning library in Go that like directly connects to Hugging Face. And I wish I remembered the name of it right now because I want to give their developer a shout out. You should have them on the show, actually.
We'll add them in the show notes for sure.
Oh, yeah. And so, yeah, machine learning has a place, but I feel like you still need the software engineers and biologists to understand each other's fields. You can't just kind of play it off on the hopes that this mushing of matrices will get the product you want, because you still have to figure out how to set up the mushing of matrices. You still have to have some concept of the data you're putting in. And the data cleaning part, as many people listening in probably know, is regardless of field, is a lot. It's like 90% of the job is the data engineering to make sure you can shove it into a matrix or shove it into some acceptable format for this to work. There's so much of that still, and I think it's still super critical, and we haven't figured out a way to generate data or manage data in a way that works to put it in these models correctly. I mean, I think we're getting better at it, obviously, but we're still working on it.
This episode is brought to you by Retool, and they have a private beta ready for you to check out. This is the fastest way to now build native mobile apps for your mobile workforce. There is no complex frameworks anymore or tedious deployments. You can build mobile apps with what you already know, like JS and SQL. This is all in the browser, no code, or what they call low code. Join the wait list. Head to retool.com/products/mobile. The link will be in the show notes. Again, retool.com/products/mobile.
From what I understand, a lot of the innovation and machine learning in the last 5 years has been around that piece you were talking about. Like, we're making sure the data is usable.
So this is a really interesting thing about biotech that's held true, is that biotech is 10 years behind on any trend, and when it comes to software. So in 5 years, I'll believe it. But right now we're still in the weeds, my man. We're still there. We're still trying our best. I mean, I'm working on a project again where we're essentially making a big graph database server thing just to have all these protein and DNA sequences and metabolic pathways in one place so you can query them and sort of engineer around it. And I'm hoping that's the innovation that's coming in 5 years for us as a field, is that we have the same amount of data cleaning that the rest, I would call it the rest of the field that doesn't rely on heavy domain expertise. Someone who has a PhD in this stuff to kind of, reason about what you're working with. I hope we can catch up, because I feel like that's just the lag. One of the things that I would say for a lot of people that are listening is egotistically, I call this Tim's law, inverse law of software quality. The more important the thing, the worse the code. And there's some, organizational psychology theory around this, but essentially the code behind Instagram is infinitely better engineered than the code behind a NASA rocket ship or your Volkswagen car. It's just how it is. And there's a couple of reasons why, but essentially if you can find some super niche thing like the operating system for a car, or maybe get the clearance to work with nuclear reactors, there's a lot of work to be done there. Nuclear reactors still run on Fortran and COBOL, man. They need some help. Bank software still runs on COBOL. I have a friend whose parents came out of retirement because they couldn't find any people to program bank software in COBOL. And so there's a lot to be done in writing sort of these niche libraries that are open source where people can see it, because the thing for me is that if I didn't make Poly open source, I wouldn't have nearly as much business. People wouldn't be able to believe in me. I don't have a PhD. That's the proof in this field is you either have to have a PhD or a corpus of work that is obviously there. And so I guess what I'd like to see in the future for Go developers is writing, scientific-esque or heavy industry software that's critical in Go or, any other language that's appropriate, that's well documented in open source. And maybe with the nuclear reactor stuff, you probably couldn't make that totally open source. I mean, the US government may have something to say about it. But I think there's some real value there, because then you start having clients come to you and go, hey, can you adapt this for our needs, our specific car? And that's kind of how I've made my career, is people go, hey, can you adapt? It's really close to what we need. Can you adapt this for my needs? And I think that's a real path to making a living as an open source developer. Not like, as a startup where you're GitLab and you're exiting for $7 billion? More than GitHub, hilariously enough. But it's a path to making a decent consulting business and a living as an mostly open source developer. Some clients they're like, hey, I just need this and I need a query for it. I'm like, cool. I'll just I'll write this pipeline and I'll keep most of it for myself and make it open source. And you get this one query that I promise never to tell anyone what this query was, and it's your data forever. Or as long as you're still a company or something. And that's totally reasonable to a lot of companies, because a lot of biotech software people they're starting to learn that they don't really have a business owning software. Owning software people think that owning software is like owning land and that it always appreciates, but no, you have to maintain it. You can't just clean it up at the end and call it a day. It's like a constant. It's kind of like surfing that you think it looks easy, right? But if you ever surfed before, I just started because I live in California. That's what everyone does here, apparently. You think, oh, the waves are gentle. Looks like you get I'm trying not to swear on this podcast. I grew up in New England, so that's all we do is we speak in swears. But you just get your butt kicked by the ocean by standing still. And that's kind of what it's like to own software. Is the waves keep coming in and you keep getting your butt kicked and you're just hoping that you can figure out how to not have your butt continually kicked by these changing tides.
No, that makes sense. So we'll do one last quick question before we do the unpopular opinion, but this is all open source, and you said that we need more software engineers in biotech. How can the community get involved in?
That's an amazing question. And so there's a bunch of issues on Poly right now, but what I truly need from engineers or software engineers right now is I don't know how to deploy large scalable things. I write really efficient DNA manipulation algorithms. That's my jam. That's what I'm really good at. But now I'm getting this data problem where we're doing this system where we have at the least 5 terabytes of legacy data that I figured out how to write a really fast parser for. But now we have to store it in a database. I'm using SurrealDB right now. I contributed to their Go client, a minor one, but still one. And it's been so far a great experience with them. And I think they have a real product going on there that's really nice and a proper graph database. So if there's someone out there that knows how to deploy things at scale that are reliable as a DevOps site reliability person, or that would be great. The other thing is I'm actually writing tutorials for Poly using Gitpod and a awesome plugin that has 10 stars on GitHub, where essentially it lets you just put in the configs breakpoints for code for the debugger. So the whole tutorial idea is that you open up this Gitpod instance and it brings you to the first tutorial, and it's just the debuggers there, and you hit run and you just go through each checkpoint or breakpoint and go, oh, this is what it looks like here. This is what it looks like here, and just keep going through that. Because I really love Go's, the Go docs example of things and that you can run them. But since a lot of the stuff is parsers and parser based, you can't really get everything you want out of that. So that's why I kind of made the tutorials for that. But I've only made the first one. There's 5 others that I would like to have that I've put in little oh, please, someone help me, write it here. So if you want to learn biology and you run it like I did with this wet lab, where I was like, hey, I want to learn biology. They're like, we'll teach you, but you got to make the class. If you want to do a similar thing, but on the internet, that would be super helpful. And if anyone's listening and you're into learning how to do the wet lab biology yourself, there are plenty of garage labs all over the world, except for Germany. I'm so sorry, Natalie. Germany has a law against this. I wish I was kidding. But the rest of the world has these. I know it's super weird, super random, but every time I talk to someone in the community who's German, they're like, I can't even pipette in my house. This is ridiculous.
Never tried.
You can try. Oh, yeah, you can. Just don't tell anyone, apparently. But there's plenty of these garage. They're called community labs or biohacker spaces. They're all over the world, and they're used to software engineers just showing up and be like, hey, what's all this biology stuff about? And, they'll take you in. They'll be nice. They'll show you the ropes. But if you want to get involved in the software, tutorials and deployment are the big things that I really, truly need help with.
And it's a good time to remind that, at least today, when we're recording the episode, it's still October, so it's still time for Hacktoberfest.
It's Hacktoberfest.
So you also get something for that contribution.
I added a tag in there, you're right. Do you get a t-shirt? What do you get for that? I've never gotten the swag. I don't
know what's this year's swag.
I have a disappointingly small amount of tech swag. I'm going to be honest, I don't have enough. And every time I get a Google hoodie or something, I just give it to my dad. He thinks it's hilarious.
So time for unpopular opinion.
Unpopular opinion. I actually think you should probably leave.
I like the harmony there. Okay. Unpopular opinion. this is probably not unpopular to people listening, but maybe it is. But open source has always been sustainable, and I see no business difference between that and closed source software, personally.
Now is the time to drop that mic.
Really? Okay. Yeah, so that's like my hot take is I don't see a difference between them business-wise. And I know a lot of people do, and it's this weird, I don't know what, there's something weird about open source in people's heads where like they hear I'm an open source developer and they're like, oh, you're a fan of Richard Stallman and you don't have proper hygiene and bada, like you don't believe in money. And it's like, maybe like two of those things are true, or maybe one. But like, the point being is that like there's this weird little stereotype of like open source can't make money. And it's this weird thing where most people, and I think most engineers, especially when they're coming from, in my realm of academia into biotech software, they don't understand that there has to be a sales team. That there has to be a person out there selling the software, getting the customers. And as a purveyor of this service, you are the expert. You are the person that like, they're not just paying for your code, they're paying for your expertise that's embodied in the code, that's in your ability to deploy it for them in meaningful and useful ways. So for me, looking at if you look at MongoDB, obviously, GitLab, and what's the other one that I've already forgotten? Red Hat, obviously. They sell services. They are the experts in these things. They help people deploy it in a way that's scalable, that probably saves them money compared to them doing it themselves. And they keep it mostly open source or open core. And the point being there is that they have sales teams. They have people going out, finding customers and doing all this other stuff. But I meet a lot of engineers who have the belief of if you build it, they will come. And for consumer level stuff, which is not what biotech is. Biotech's very business to business. I mean, like there are some consumer biotech things as end products. And there are some people that, are consumers as students or people that buy like home kits or things like that. But, it's a very business to business thing where like my code, like being open source, people ask me, aren't you afraid if someone's going to copy your code? It's like, no, like I'm the guy who wrote it. Like, sure, you could use my code, but like, why would you pay someone twice as much to do half as much with it as I would do if you can just hire me? And I think that's something that a lot of people don't recognize with open source stuff is that it's a very viable, it has almost no effect on their actual business model. That's what's always been surprising to me. Unless your business model is to not develop something novel, maybe you're like, a lot of SaaS is obviously like, different projects, just a couple mess of open source packages put together in some sort of microservice framework that blah, blah, blah. And like, obviously, sometimes it's novel stuff out of that. And sometimes it's just reskinning of other people's open source stuff, but like a fancier GUI or something.
Yeah.
But if you're writing something truly novel that no one else has done before and you're the expert, you really have no fear of anyone copying you because you have to market yourself. That's the big thing. You can't be the quiet guy in the corner. You can't, be like, oh, I wrote this thing. And then you let some super marketing guy just come in and be like, this is my thing now. I made it, blah, blah. You can't let them do that. You have to have some hold there. But I don't see much of a difference between a company whose product is open source and who's not. It's still the same thing in the most part. But you do get the benefit as an open source company as people can believe that your software is good because they can literally see it. Like they can see, the proof is in the pudding there. They can look at it. And, with a lot of biotech software, people, they're like, just believe me that it's good. Please give me $100,000 to start before we even show you the code. Just believe me. And so, there's a big difference there. And then you get into, there's obvious code quality. Then you have people that are submitting pull requests and little bug fixes and little documentation fixes that, it's not like you have a real engineering force behind that, but it does add up. Like it does give you also some marketing power where like now people are like, hey, have you heard about word of mouth can kind of happen there a little bit. Like I show up to meetups here in the Bay Area and people are like, oh yeah, you're the guy who wrote Polymerase. I was like, nice to meet you. Like, so there is some real value to it, especially if you're not like the traditional purveyor of truth, if you're not the PhD who's gone to Harvard, who's the fancy guys, grad student. It's a great way to show that you're the best and you've got the right stuff. And like, so I feel like there's going to be more companies, hopefully like GitLab, Red Hat, MongoDB, and I'm hoping companies like Gitpod and Nextflow sort of get to this IPO stage too, where like people can see the engineering, they can see the, the service, they can see the value in it, and they don't care that it's open source because they know that it would cost them $60K to have a dev do it internally, or it could cost them 5 to 10 to hire the guys who wrote it. And I think that's just sort of, I hate to use VC terminology. I've been spoiled by the city. That's the moat, quote unquote, is that you're the moat. You're the creator. You're the person who made this. You are the artist who brought it to life. And there's some real value behind that.
I agree with you very much.
Really? Okay.
Yeah.
So it's not controversial, and I ruined the segment.
I think it might be controversial.
Yeah. So there will be a vote, as with every unpopular opinion. We will put this on our Twitter and we'll see how many people agree or disagree with you. But to summarize it, we said that your unpopular opinion is that open source always has been sustainable. And yeah, we'll see how that goes. And
Yep.
I have to say, this must be the episode with the most show notes we will have, at least for me.
Oh God, I'm so. Oh.
No, that's amazing. You taught us so much, and I want to say thank you very much for that.
I wish I had a whiteboard. I'm gonna have to go and make a new PowerPoint presentation.
I learned 1 million things today.
Okay, all right. If anyone is out there listening to this, or you, Natalie, or Ian, like want to ask more questions about biology, I'm always available. I may get tired of it eventually.
And your Twitter is mentioned here for sure.
Oh yeah, there's the Twitter, there's the Discord. If you jump in the Discord, there's gonna be hopefully some nerd that'll be like, I know that bit of biology. I love my Discord for that. Someone will be like, yeah, I can explain that. I'm like, great, I had no idea.
Perfect. We'll add the Discord to the show notes as well. Ian, thank you so much for joining and co-learning here live. Tim, thank you for teaching us so many things. This was really fascinating.
It was.
Thank you for having me. I hope I did all right.
It was amazing.
Amazing, yeah.
All right.
Thanks everyone who joined.
Thank you very much. Talk to you later. Bye.
All right, that is the show for this week. Thanks for hanging with us. If this is your first time listening, subscribe to the pod at gotime.fm. And if you're a longtime Go-Timer, do us a solid by sharing the show with your friends. Help us help more people with weekly Go-related goodness. Thanks once again to Fastly and Fly.io for partnering with us. Check out what they're up to at fastly.com and at fly.io. And of course, thank you to our beat freak in residence, Breakmaster Cylinder. Our beats are banging because BMC bangs out dope beats. That's how it works. Next time on Go Time, Natalie and Ian team up again. This time they're talking debugging Go programs with Laurent Haimovich and Tiago Quiroz. Stay tuned for that. We'll have it ready for you next week.